Good morning, my name is Pablo Morón Elorza, and today I will present our poster entitled Capillary Zone Plasma Protein Electrophoresis Analysis in the Nussbaum's arc, Silurianus stellaris, maintained under human care. So plasma protein electrophoresis is a test that measures specific proteins in blood plasma. This test separates proteins in the blood based on their electrical charge and size. Electrophoresis has been demonstrated to be a useful clinical tool in human medicine, companion animals, and recently in non-domestic animals, including Elasmobranchs. In humans and most mammal plasma protein electrophoretograms, we can distinguish 5 main fractions from left to right, which would be albumin, alpha-1, alpha-2, beta, and gamma globulins. In Elasmobranchs, interspecific variations are great, and to facilitate protein electrophoresis interpretations, electrophoretograms have been previously divided into 5 different fractions. The objective of this study was to perform plasma protein electrophoresis analysis using capillary zone electrophoresis in the Nussbaum's arc, a species belonging to the order Carcharodontiformes, which is distributed in the northeastern Atlantic Ocean and the Mediterranean Sea, and is considered vulnerable by the IUCN Red List, with a decreasing population trend. For this study, we collected samples from 62 clinically healthy individuals, being 30 females, 32 male, 23 subadult, and 39 adult. Blood samples were centrifuged to separate plasma, which was frozen at -18°C until processing. Processing was performed using a microcapillary minicap protein 6 flex piercing, and plasma was previously diluted at 140 dilution with capillaries urine dialysis buffer following manufacturers' recommendations. So what we found is that capillary zone electrophoresis allowed the division for the electrophoretrogram fraction into 8 consistent fractions in the Nussbaum's arc, which are represented here. We also found statistically significant differences between sexes for fraction 4, and between developmental stages for fractions 5, 6, and 8. As a poster, there is not much time to discuss the obtained results, so we can conclude that the plasma protein electrophoresis baseline values provided in this study will aid in the Nussbaum's arc, Silurianus stellaris clinical evaluation and health management. Although further studies are warranted to completely understand the composition and clinical implications of the different fractions determined by capillary zone electrophoresis, the possibility of cutting the electrophoretrogram into several consistent fractions will allow for a more accurate interpretation of physiological and pathological variations. These are the references used to develop this presentation, and thank you all very much for your attention.